Good morning and welcome everyone to today's Critical Path Institute Rare and Orphan Disease Programs webinar series. We're going to give it just a minute to allow everyone time to filter in, and then we're going to get started. Okay. Once again, welcome to today's Critical Path Institute Rare and Orphan Disease Program webinar series hosted by RDCA-DAP. Just a few welcome and housekeeping things I'd like to review before I turn it over to Heidi Grabenstoffer to introduce our speaker today. So first of all, if you have questions that you'd like to ask our speakers or panelists, please feel welcome to put them in the Q&A chat box at any time. Please make sure to send it to all panelists to make sure that we see them coming through. We will answer those questions when we get to the panel portion of today's webinar. Additionally, all of our lines are muted just to reduce background noise. So once again, if you have questions, go ahead and put them into the Q&A box. This webinar is being recorded so that we can make it available on the CPath YouTube channel and our website after this call. And if you have any questions regarding Critical Path Institute, the Rare and Orphan Disease programs, this webinar, the topic, questions for the speakers that we don't address today, please feel welcome to email RDCA-DAP@cpath.org. You can see the email address on here, and we'll make sure to follow up with you after this call. So with that, I would like to go ahead and hand it over to Heidi Grabenstoffer, the Scientific Director for RDCA-DAP, to welcome our speaker today. Thank you.
Good morning, everyone. It's my pleasure to introduce Laurent Servais. He's going to give us an overview of the Actimyo device that he has developed with patient advocacy groups and talk to us a bit about the use in neuromuscular disorders and the use in potentially in non-neuromuscular disorders. Laurent is a professor of pediatric neuromuscular diseases at the University of Oxford in the UK and an invited professor at the University of Liège in Belgium. He graduated in medicine and pediatrics from the University of Louvain in Brussels, and then trained as a child neurologist in Robert Debré Hospital in Paris, France, and as a myologist in the Institute of Myology at the University Hospital in Paris. Professor Servais leads the pioneering genomic NBS program, Baby Detect, that aims to screen at birth all treatable and severe conditions. His other research interest covers innovative digital outcome measures derived from magneto-inertial sensors, which we'll be hearing about today. Next slide, please, Tanisha. All right, then we'll throw it over to Laurent and introduce the additional panelists after he speaks.
Hello, everyone. I guess you can hear me. I'm going to hopefully share my screen. Can you see my screen? Obviously, you can. Okay,
we
great. Fantastic. So thank you very much for hosting me today. I'm very happy to present you the results of 14 years of work with not only patients advocacy group, but also I'll say all the community, all the stakeholders, brilliant engineers from CISNAF, pharma companies, and one of them being very involved, which is Roche. But patients advocacy group, patients themselves, regulators, ethic committees, and maybe it's the first take-home message from this talk is that we need to work together if we want to achieve this type of qualification. So I will first try to describe the story of the qualification of the SV95C and to explain hopefully what it is, and then to try to figure out how we can use it as a kind of breakthrough in the field of digital biomarker. I have a couple of disclosures. So the main problem that we are facing, and I can tell you my personal history, is I was in Paris as a young myologist, and I was looking at a patient walking in a 6-minute walking test, and that day the patient was not very good. And the reason why it was not very good is that the patients, they can be different one day to another, and they can have good days and bad days. If this is the evolution of two groups of patients or two patients, obviously they are diverging, right? But according to when you consider the last measure, you could end up by concluding that these patients are not different, but they are, because if you could consider the area under the curve rather than just a point assessment, you will conclude that indeed this two group of patients are different, right? So being able to have a continuous assessment rather than a point assessment could potentially be more powerful. And that's the Eiffel Tower syndrome. So in Paris, this patient was not going that well. The mom told me, oh, the problem is that yesterday we went to the Eiffel Tower. We visited Eiffel Tower, so he's exhausted. And so he was down performing. And we were actually gambling decades of research and investment and in a phase 3 trial in a patient who was tired because of he visited the Eiffel Tower. And we considering rare disease, right? Patients in a rare disease trial, they come from far away, right? And they come to the capital because the sites, the investigation sites are in the capital. So when they are in Paris, they visit the Eiffel Tower, and when they finish the Eiffel Tower, they go to Euro Disney. But this is an example. So how can we fix it? Well, the first way is to do the clinical trials in Brussels, because you will never be exhausted with the mannequin piece in Brussels, which is a very small and lovely statue. You will never be tired, but it's not, you cannot conduct all clinical trials in Brussels. So we need another solution, and definitely wearable devices is the sense of history, right? Is the only question is not if we will use wearable device. The question is, how long do we need to really accept it as a concept that could lead to drug approval? And this was last year, the increasing number of clinical trials using wearable device, and you know that the FDA has funded the Digital Health Center for Excellence. So the regulators themselves understand the importance of wearable device, and this is the example for 3 different conditions in neurology, like Parkinson's disease, multiple sclerosis, Alzheimer's disease. As you can see, there is an increasing number of trials using wearable technology, especially when it comes to motor tracking. So this is the use of these different wearable devices according to what you try to measure, sleep or cognition, speech, but motor tracking is definitely the main indication of using wearable technology. A little bit of self-promotion. This is a work from my PhD student. She conducted a large systematic review on the use of wearable technology in rare conditions, and she found a very large number of studies, including 3,600 patients. So yes, it's increasing. Yes, we use digital technology and in more and more conditions and in more and more trials. This wearable, you can wear it wherever you want, basically. And this is the figures from this studies. And as you can see, the number of sensors goes from 1 to 6. So you have plenty of different configurations. So the question is, we are using more and more wearable technology, and wearable technology is not something from the 21st century. It's already something from the 20th century. And there are a large number of trials, but at the end of the day, how many digital outcome do we have that are qualified as a primary endpoint, and how many drugs are approved on the basis of a digital technology? And the answer is not so many. And the question is why? So I told about the Eiffel Tower syndrome. I told you about the Christmas gift syndrome. The typical, I suffered of the Christmas gift syndrome. What does it mean? I mean, you are a clinical trialist and it's Christmas and your mom offers you a beautiful wearable device. Probably because you become fatty, you don't work so much. So you receive a nice tool that is going to help you to move more. Okay, fair enough. And then you say, well, I come to clinical trial. What can I do with that, right? And the short answer is nothing, because it's not fit for purpose. It's not a device that is done to, and it has been conceived to measure a small change in patients over a long period of time. And especially the fact that, it's most of the time not very well calibrated for temperature, so that when you go in and out quickly, that the device is into the wild. So at the end of the day, if you look at plenty of publications, it's exactly a Christmas gift syndrome problem. Is that it's about a out-of-the-shell device, and some patients, and well, we can see that the patients are different of the controls. Okay, great, but where does it go? Most of the time, no. So after having suffered of the Christmas gift syndrome, I met the engineers and we had a very nice discussion. The engineers really wanted to collaborate with the doctors. They say, well, I will offer you the moon. I say, okay, fair enough. The moon is great, but I would prefer an alter of movement. So what do you mean exactly by an alter of movement? Well, I would like to be able to identify all the movements of the patients, right? And then when this movement has been identified one by one, to quantify them very precisely, and in an uncontrolled environment, right? And then during 2 years at least. And I don't want to have a shift during this time period. So I want that whatever I measure at the beginning is the same as what I measure at the end. And of course, I need to be compliant with all regulations, not only the one in my country, but any patients around the world. And I want the internet transmission, of course. And then the engineers understood that the moon was probably a good deal in comparison, because indeed it took quite a long time. It started in 2010, so it means that we were not very rapid, and we started with sensors to try to evaluate what are the right sensors to measure and what we wanted exactly in terms of precision. So we made a prototype that was, not wireless, just to identify the right sensors. We moved to a second prototype that was absolutely ugly, and it was actually Bluetooth. So the battery were just insanely big. And so then we went to a third prototype that was awful, and then to a fourth prototype that was equally awful, but it was a medical device. It was in 2017, and from there we could extract a first outcome, which is the SV95C. I'm going to discuss what it is, and get to the qualification as a secondary outcome. And then we went to a prototype that was slightly better designed and more acceptable, and then getting the qualification as a primary endpoint. So just to explain you that the system of thinking was much more to starting, what do I need, and then to construct the tool that I need, rather than trying to take something off the shell and say, well, maybe it will help. At least it's beautiful. So we made something beautiful, but at the end. And I think it's the way to do, but the problem is that you may exhaust the community, because obviously, as you might imagine, you iterate a lot with different versions. They are ugly, they are not, well designed, they look like something that people wear when they go out of jail. So it can really, at one point jeopardize the development to move along these lines of getting something beautiful, but only at the end. So that's the last version of the site, which is actually the device that we use. Okay, it's quite small nowadays. It's smaller than an Apple Watch, and with a weight of 25 grams. So it was much more burdensome. The Actimyo, this new version, is much smaller. And typically, if you wear it at the ankle level, this is what you can measure. So this is a stride. Patient's walking, then the patient falls, and then the patient stands up, and then he continue walking. So as you can see, we can identify every single stride, and we can measure every single stride. That's what we wanted to do, to have for the lower limb, but we can do the same at the upper limb. Really the possibility to identify every single movement and to measure every single movement. So cool. So the patients wear it during the day, the device goes on a docking station during the night, and then the data are transferred by the cloud. And then we can identify the strides, measure the strides, having a distribution of the strides, and then to consider the 5% most rapid. This is the stride velocity 95th centile. Okay. The problem is that the variability. First problem. When I was in French, I had this theorem in France. When the French are on strike, they walk more, obviously, because there is no public transport, right? Then I moved to the UK and I changed my mind. Say when it pours, British people walk less, and when it simply rains, they walk more. It means that our activity is weather dependent, right? So that's quite a problem because the weather may change. And if this is a kid, he lives with his mom, and then he goes to the cousins, the mom side, and then he goes to dad, who has just an interest in his car. And then the cousins, the father side, as you might see, the variability on a day-to-day basis is huge. But first thing, if you average on 3 days, you will decrease the variability. And if you average on 9 days, you will decrease the variability. And if you average on 2 weeks, you will even decrease more the variability. So this is actually a fact that the variability depends on how long you record the signal. That's in mathematics what we call the Allen variance. It's and it depends not only of the social variability, but also the variability of the disease. If you have a disease that doesn't change over 1 year, the longer you record, the better it is. If you have a disease in which patients decline, if you actually record a longer time period, then you include, of course, period of time during the patient may potentially be weaker. So that's for the stride velocity. And as you can see, this is a logarithmic scale. When you record the patient during 180 hours, you get a standard deviation of 3%, which is not that bad. I mean, 3% in a clinical trial, I'm quite happy with that. If I conduct a 6-minute walking test, I would never have 3% of variability. So we are in an uncontrolled setting, but nevertheless, we can be much more precise than in a controlled setting. And the second thing is that it's not only about the duration, but also whatever you measure. What does it mean? If you measure the total number of strides, this will be really weather dependent, because if it rains during 2 weeks, like cats and dogs in the UK, you don't walk so much, right? But if you just consider the maximum velocity, which is the SV95C, sooner or later during these 2 weeks, you will have the opportunity to do it, right? Especially if you're a child, which means that another way to tackle the problem of variability is not only to have longer recording, but also to go to maximal velocity rather than cumulative data. And then why should we go to the 95th centile stride velocity? It's actually because this was the centile for which we had the best size effect, which means the larger variation in patients with a smaller standard deviation. It's actually not dependent of the compliance. So if the patients are highly compliant, it doesn't change anything. Of course, it needs to be correlate with the other outcome, like the 6-minute walking test, and you need to be discriminant with controls, with healthy controls. This is the correlation with different gold standard in Duchenne muscular dystrophy. this is how it declines with time in Duchenne muscular dystrophy. And then for EMA, a very important point is the MCID. What is the minimally clinically important difference? And this is tricky because obviously there are different ways to measure this, to calculate the MCID. You may have what we call population-based approach. You can have anchor-based approach. And don't believe that whatever is the way you will reach the same the same number. This is not true. Obviously, it's very different. So it's tricky because the MCID will be critically important in a clinical trial. If the MCID is very large, it means that you will need to have a major effect of your drug to reach the MCID. So we need to be very careful in this definition. This is the discrimination with controls, and that's of course critically important. And again, the sensitivity to negative change helps you to calculate how many patients you need to conduct a trial, right? But then when we were at the EMA told us, well, you have the sensitivity to negative change. That's fantastic. But do you have the sensitivity to positive change? And I say, oh, that's a good idea. It's a good point. But the problem is that there is no drug that is approved in Duchenne. So in conditions where there is no drug that is approved, and because you don't have the right outcome, and to qualify the right outcome, you need an effect that works. It's it's a little bit like a vicious circle. So we could demonstrate that we could not only use a smaller sample size of patients, but also a shorter period of time to observe the same effect. So this is the long and winding road of the qualification as a secondary endpoint. And EMA, we started in 2016, and it took 3 years, 3 years of discussion. And just to say that they probably today we will do it much faster, hopefully, but don't expect this to be rapid. This is not a rapid process, and this was only EMA. We came back in 2021, and within 2 years, we could get the qualification as a primary endpoint. So the level of the discussion dramatically changed between the secondary and the primary qualification. Obviously, EMA was much more used about this type of approach. The questions were much more to the point and much more nasty, to be honest, and mostly focused on the MCID, which was a key point definitely for the EMA. So this is especially when it comes to the primary qualification, a big piece of data, a big cake. That's what I call the ambulant patients between 4 and 15 years old. But then I wanted to, and we wanted as a team to get the cherry on the cake. And the cherry on the cake for me are the younger patients, the patients 2 to 4 years old. And the reason is that these patients are very difficult to assess, right? If you ask to a 3 years old to go for a scale or to make a test, it's quite complicated. So we included, today we have 27 patients younger than 4 years. This is the first analysis in a couple of patients, 12 patients, 2 to 4 years old and 10 controls. And what we could show is that the toddlers, let's call them the toddlers, are very compliant. They don't care actually to have a watch, right? Especially if you explain that it's a super power watch or whatsoever. The reliability in toddlers is insane. They have a reliability of 0.96, right? It's huge. And the reality is that actually, if you ask a toddler to do something, maybe he will do it. If you ask him to do something the way you want him to do something, probably he will not do it. And if you ask him to do it again, you will never get the same things. But if you place a sensor on a toddler and you tell the toddler, okay, live your life. I don't care. It's, he will, during 1 month, live his life, and he will at one point make his top performance, right? And 1 month later, if you place the sensor on the toddler again and you tell the toddler, I don't care, do what you want, he will do pretty the same, and he will at one point make his top performance. So now my PhD students used to tell me, okay, if you want me to be reliable and to express my top performance, let me do whatever I want. Well, it works for the toddler, but it doesn't work for the PhD students. The beauty also is that there is a very good discrimination between controls and patients, even at the age of 2 years, which means that it really opens the possibility now to conduct trials in much younger patients. So it was just published after the qualification in Nature Medicine. And then for those who are in the field of neuromuscular disease, you could potentially see that a very expected trial in gene therapy did not reach the primary endpoint for the which was the North Star, but there was a significant difference as a secondary endpoint of this SV95C. The three together was quite a big start for this type of approach. So now we consider much more broadly if we have these mountains of data that we extract from patients, we could not only extract the symptoms like the power, but also to start measuring or trying to measure the fatigue and the activity, the falls, the ataxia, the abnormal movement, not only to have a signal of action of the drug or having a primary endpoint in a pivotal trial, but also to convince the payers or to convince the patients themselves that the drug helps them. So this Holy Grail, which is the SV95C in DMD, could we find other example in other conditions? So we developed two protocols that we call Actimyo and Acti-Liège, and they are umbrella protocol. What does it mean? It means that now when we want to develop in a condition like Angelman, like CTNNB1, we just invite the patients in one of these protocol in first Actimyo, and we try to measure and to be sure that when we speak about a stride of 90 centimeters, it's actually 90 centimeters. So, for instance, this is a lovely girl with a Angelman. What can we objectively quantify, right? What can we measure? And we tested different type of measures, not only in the controlled environment, but also in the uncontrolled environment, to figure out what was exactly not only the variable that we were able to measure, but those that were reliable, that were discriminant with controls, discriminant between different types of genotypes. So two approaches. The first one is the clinical validity, the analytical validity, and then the clinical validity. So that's the first step when we measure actually with a motion capture and with an Actimyo or a side. And the key question is when a patient makes a movement that we consider to be 30 centimeters, is it really 30 centimeters? So it's the analytical validity. And then when we are confident enough that we measure the right thing, then to have the patients in an uncontrolled environment. And then the question is, can we really measure that in an uncontrolled environment when there is the subway, the car, the train? What is the compliance? What is the reliability? What is the optimal recording period for this condition? What is the external validation? And what is the natural history of the disease? And nowadays we're conducting several development in Parkinson, in MS, Angelman, CMT, FSHD, SMA, Huntington, ALS, MG, and CTNNB1. We will have the opportunity to discuss it. So it's over 70 clinical trials, 2 million of hours of recording and more than 30 sponsors. So the take-home message is that now digital outcome has made its way in the regulatory landscape as primary endpoint. And it has the potential to dramatically reduce the duration of clinical trials and the number of patients. Now we need to think disease per disease, how can we apply this type of technology? And I give just the example of magnetoinertial sensors, but this can be also used in many other approaches, right? If you want to ask me as a question, what are the 4 learnings over the last years in this process, I would say that one, the sensor is key. If you want to go to the granularity of individual movements, you need to have an excellent sensor. And I think that it's a nonsense to try to record with any sensors and saying it's you can be sensor agnostic. I'm strongly convinced this is not true. If the objective at the end of the day is to conduct trials with much less patients and in a shorter period of time, we need really to pay attention of the quality of what we record. The second thing is that data is key. Is then and to acquire data, it's very difficult because you need to do it the proper way. You need also to have the clinical data that goes with, and you need to invest a significant amount of time, energy, and money to acquire the data. The third thing is that having a long-term plan for development of an outcome in a disease is key. It means that what you will be asked by FDA or by EMA is different. Do you need qualification? Maybe not. Do you wish to get the qualification in both agencies? Maybe or maybe not. But according to the strategy, you will really need to have a plan because the process in the different agencies is very different. So so the priority will be different also. So it's quite important not to do what we've done, which means to say, okay, let's start with, I don't know, EMA because it's closer. Well, you know, probably have a better planning is important. And the fourth and maybe more important point is that the interaction with the physicians and the engineers is key. And this is a typical discussion between, on the right side, a physician, and on the left side, an engineer. So it was a typical discussion that we had over years with the engineers explaining, you know, very clearly what is important for them, and the physician explaining what is important for his patients. And sometimes it's a complete, you know, nonsense discussion. So I just want to finish by thanking my team, especially Margot Poller, with a PhD student very involved, to thank Roche, who is a partner, an important partner in this project, the brilliant teams of engineers in CISNAF, and one lady here was being super important in this process, and unfortunately who left us last year, and we are all extremely grateful for all the work she has done. Thank you so much for your attention, and I'm more than happy now to have this open discussion with the other panelists.
Thank you, Laurent. That was wonderful. I would like to take this opportunity to introduce our esteemed panelists of patient advocacy leaders that will now have the opportunity to share some of their experiences working on developing the Actimyo, given the different complexities of this disorders that they work with. And to start with, Alison Berendt, DVM and DACVIM, is the Chief Scientific Officer of the Foundation of Angelman Syndrome Therapeutics and the co-director of Angelman Syndrome Biomarker and Outcome Measure Consortium. Eric Camino is the Vice President of Research and Clinical Innovation for the Parent Project Muscular Dystrophy Foundation, and Spela Mirozevic is the co-founder and president of the CTNNB1 Foundation. We also are being joined by Sakshi Sardar, who is the Senior Director of Digital and Precision Medicine at CPAC. I'd like to ask each of the patient advocacy leaders to share a little bit about their experiences thus far. You're kind of on different developmental paths and also have different symptoms with your disorders. And I think it would be educational for all of us to learn how you've approached development with this DHT. Eric, would you mind if I started with you?
Yeah, absolutely. So I think, as Laurent kind of covered, there's been a lot of work done in the Duchenne space with 95th percentile. And I think as a field, we're seeing more of kind of an uptake of it within our industry partners, which is great. The qualification with Europe is great. And hopefully with the FDA, just to give confidence to drug developers as they continue. And I do think it's really important that we work and develop these more sensitive measures. One of the challenges within Duchenne, as Laurent alluded to, is the trouble with getting approvals. And a lot of the approvals we have in the US are based on accelerated approval. We have limited approvals based on functional outcome measures. And the one very recent case we did required an 18-month study. And had that been stopped at our kind of traditional 48 weeks, the sensitivity of our current outcome measures wouldn't have shown efficacy. And so having a device that kind of breaks from this snapshot capture within clinical trials can help us combat some of these longer windows of time to capture functional change in patients is really important and something that we need to continue to push to kind of drive the field forward. And so happy to see it being taken up by companies, included in a lot as secondary and exploratory outcome measures. And then the other piece that I think Laurent touched on, which is really important right now for Duchenne, is we're starting to see newborn screening implemented in different states in the US. So New York, Ohio, Minnesota, and potentially added to the RUSP, we hope in the near future. And so as we start to identify babies, we don't want to have to wait until we get to our kind of traditional paradigm of 4, 5, and 6 years old to start enrolling them in clinical trials and understanding if we can have early interventions that help affect the trajectory of the disease. So having an outcome measure that can help us in these younger populations is going to be really critical to earlier intervention and hopefully kind of transforming patients' lives.
And to piggyback on what you said, as a digitally derived endpoint, one of the audience members asked, what I'm trying to find the specific question, but they were fascinated by the choice of the SV95C and curious on how patient experience data and patient insights may have contributed to the choice of the SV95C for the development of the measure, and to point that to both you and Laurent, following everything that you just said.
Yeah, and so I can say that there was some work that was done with Sysnav, the company that was developing the Actimyo, with a number of patient organizations kind of globally, where we did some survey work with them to try and assess with the patient community to understand that meaningfulness, and looking at something like walking ability as something, very important to our community. Obviously, loss of ambulation being one of the big milestones. So that there has been some work underway. I'm sure Laurent probably, being closer to it can provide some additional detail.
Yeah, so indeed, there was a formal and informal interactions with the patients. And I think the formal is important, but the informal is very important also. Everything started, of course, during clinical trial and with interaction with patients and physicians to figure out what is important, because we thought that capturing the data on the continuously made sense, but we had no idea of what to measure. And so the informal interactions with patients were extremely important. And then, of course, when it comes to the regulatory discussion, you need to have a formal understanding of what you measure, and to show, to demonstrate that what you measure makes sense for the patients. And so we had we made this exercise in Duchenne. We made this exercise in neuromuscular disease, and we made this exercise in Angelman, and this can be a nice way to hand over to Alison, who has been instrumental into it. obviously it's not such an easy question because it's a much more complex condition. So yes, this is needed, and this is needed especially at the FDA level, because believe it or not, for EMA it's important, but EMA is much less picky and nasty than FDA on that. For FDA it is the beginning, even before the qualification package plan. You need to demonstrate that what you measure makes sense for the patients, which can be challenging, by the way, because at the beginning sometimes you have not yet a clear idea of what you measure, but nevertheless that is the very first step for FDA.
Allison, with that, since Laurent suggested it would be a great transition, would you like to share a little bit about your experience?
Sure, happy to. Thanks for having me. I would say that, in a disorder like Angelman syndrome and other neurodevelopmental disorders that are not degenerative, I think you're looking at this from a very different perspective. Ultimately, we're not looking at these individuals as ones in which we're trying to maintain motor function or ensure that we even potentially regain motor function, because our children never lose their motor function. They just have an abnormal gait, and that abnormal gait is associated with significant ataxia, incoordination, falls, and the inability to ambulate in a way not only that's safe, but that doesn't require adult support, or that ultimately doesn't allow them to have a duration of gait in which they can be functional, like going to the store with their family member without needing a wheelchair or pushchair, which due to the fact that they may not have the endurance to do it, they may not have the gait function to be able to navigate obstacles and navigate things in their way in an everyday setting, or they may actually have a lot of falls, in which requires an adult to be with them at all times, so that adult can't be shopping in the grocery store while holding the hand of the individual. With Angelman syndrome, we're in a little bit of a different situation on what we're exploring as far as what's meaningful, and then what, of course, is measurable for this population. And I think this much more models a lot of the neurodevelopmental disorders in which gait disturbance is a major part of the symptomatic domains that are important and meaningful to families. And so having something objective with digital health technology that you can actually measure objectively in a real world setting is incredibly important. If you put a child in a hospital setting where they're maybe doing some of the objective measures that are standardized for neurodevelopmental disorders, like the Bayley scale of infant and toddler development, for example, the big issue here is that in a hospital setting, no one's going to let a child fall. Everyone's going to be holding their hand and being very cautious of what they're touching in a hospital setting and who they're going near. And you can't demand a child with any type of cognitive delay to walk 6 meters, or to walk straight on a line, or to turn around and walk back to you and then objectively look at their gait patterns and their gait quality. These are children that have much more global delay and much more global impact, and therefore you need to be able to measure something that isn't actually measuring their cognition, but is actually measuring their gait function. And that is very hard to do if you don't have something like this device, because what we can measure is their ability in everyday setting to walk, and to walk for a duration of time that is more meaningful and impactful to families, their ability to walk more safely, to navigate obstacles, and to be able to measure the amount of times they fall. Falls are a major, major issue for kids with Angelman syndrome and other neurodevelopmental disorders. And so we're not, what's meaningful and important to our families is really safety. And it's also the burden of abnormal gait that has on not only the individual, but their families. So if an individual can walk safer, or can navigate stairs, or can navigate to the restroom, that is going to change their life in a hugely meaningful way that is not necessarily degenerative, that they're not losing those skills. They never had those skills. And so we're looking for them to be able to gain those skills over time. And there's very little ways objectively to measure that that's not in an in-home setting. Which certainly on things like the Bayley, you're going to absolutely miss that granularity. You don't gain a single point on the Bayley if you don't go from walking to running. And there's a lot of important things between walking and running that will change the lives of individuals that you will never get on something like the Bayley. And so you really need to be able to look at that granularity, understand the meaningfulness of that granularity, and then figure out how you're going to measure that. And this device does seem to be able to capture all of those things for these individuals.
Thank you. I mean, well said that small changes are meaningful for everyday life for families caring for children with neurodevelopmental disorders. There is a question about how one can engage you to use this for a clinical trial, and I'm assuming it's from a trialist or a industry partner. And they're screaming your accolades on what a wonderful device it is. So Laurent, I'll give you an opportunity to address that, and then I'd like to allow Spella some time to talk about her experiences.
So, as you might imagine, I'm not a device manufacturer, so I don't have a lab with constructing devices. So this is done by a company, and I need to say I have no personal interest into it. So the company is called CISNAF. So they are pretty flexible. So according if you are an academic, if you are a pharma, and what you need exactly. So the best way is to drop them an email, SYSNAV, and I can certainly write in the chat some contact name from this company.
So as we're coming up on time, I'd like Spella to please share a bit more about your disorder and the experiences you had working with Laurent.
Hello, thank you very much for having me. It's I think really great opportunity for our rare syndrome, as we currently only have 430 patients registered via our community. We are, I think, the youngest in this journey with this device, but not less enthusiastic. A few of our children traveled to Liège to test this device in a controlled environment a few months ago. To be honest, I was a bit concerned, especially because our disease is neurodevelopmental and our children experience severe behavioral problems. And I was especially concerned whether my son would be able to have it in his hands and legs, or would he be just, screaming and be, just excluded from this protocol. I was pleasantly surprised. I was able to distract him. And even when we came home, I bought these things in the store to put him on his arms and legs to practice. After a while, he was able to adapt it. And it's, I think, really good because even, it's an information that even in severely behaviorally affected children, there is, this device can also be applicable in such a in these children. And so now in 2 weeks, we are organizing our second International CTNLB1 conference, and 20 children will receive this device, take it at home, and hopefully we will be able to receive a variable that will be specific for CTNLB1. And I think this is a really great opportunity for our syndrome because we don't have any, or we haven't found any biomarker. And so currently we are relying only on, the Bailey scale or other patient-reported or family-reported questionnaires that we know are not, as objective or even as sensitive as we would want. So I'm very happy. Also, I would like to comment, for example, as opposed to Angelman syndrome, in our children, for example, only 30% of children can walk. And that was, also one of my concern. How do we choose children for a clinical trial? For example, we will choose only those who walk. And, what does then happen with those who do not walk? But I do see this device also more as a proof of concept. I've heard comments like, in your disease is going to be really hard to see any effect. It's not degenerative, and unless the treatment is super effective, it's going to be really hard to see an effect. And so having this device to support seeing sooner and seeing maybe even smaller but yet meaningful effect is very, very important. Only just one last thing. I recently engaged with our community asking them what is the minimal meaningful outcome they would like to see in any kind of a clinical trial or any kind of treatment in the future. And without a breath or a second, they said, we would take anything. Anything is meaningful. We just want the disease to stop because we do see some spasticity, or just to improve a little. We would take anything. Thank you.
Thank you, Spell. Sakshi, I was wondering, there's a question that has come in, and Laurent, if you have any input. Sakshi works with our neurodegenerative consortium. They're asking what efforts are underway to establish common protocols and data formats across DHTs that may contribute to DHT-based healthcare systems. Is this something that you are familiar with?
I am actually a little bit familiar with. I think it's a very nice vision. The reality is that for me, we should not consider that DHTs are just interchangeable, and that you can say, well, whatever has been collected was one is the same than the other. I mean, sensors are different, calibration are different. I mean, we cannot just say that for me, as I say, that the sensors and the quality of the sensors makes a lot of sense. So there's been effort, especially through a consortium, EMI consortium, to evaluate the how agnostic you can be for the device. The question is, what level of precision do you expect from the device that you are comparing? Because if you are comparing devices that allows you to give you a rough estimation of the patient's activity, probably you can easily interchange change the different device together. But if you want to go a little bit deeper in the individual movement analysis, then the precision of the sensors become key. So short answer is yes, but not so much by the device manufacturer. It's more a kind of academic and also pharma-driven effort. And B, to the extent of the precision that you want to measure. And if you accept to remain quite broad level, if it's okay. If you want to go in the precise evaluation of the movement, the sensor is too important for that.
Thank you, Laurent. I think this is an unmet need that is vocalized very often. Are there efforts for standardizing some of the protocols? I believe there are some efforts from CDISC on that side, and additionally, there was a work done by the 3DT effort at Critical Path Institute, where they developed the metadata-based framework that can help in establishing some of the requirements associated with protocols so that it can be contextualized when it comes to utilizing those trials for drug development tools. Those are some efforts that I'm aware of. But it's definitely a very important need that we should think about.
Yeah, certainly. But we need also to, and I agree with you on the principle, but we need also to figure out that all of this comes, of course, with IP, right? And we need to remain pragmatic. This is not a very bankable market. I mean, if you compare with the drug market, if you are an investor, you will just stay aside of DHT and put your money into a drug company. You will get a much better return. So the the problem is also to try to find the right incentive, because all of this, I mean, you've seen the process. I mean, for me, it's okay because I'm an academic, right? It's my project. But when it comes to bring industrials and to pay for engineers and quality control and device, this costs a lot of money. And the market behind is not big. When you speak about Duchenne muscular dystrophy trials, it's not an enormous market, right? So I think it's good to try to figure out how we can use one or another device or interact. But the problem is that it comes with a very pragmatic questions of keeping this as a potentially attractive market for investors and for people who wants to develop. And this is a balance that is not easy to find. Again, speaking as an academic, which is in the middle of the game, right? But we need to be conscious of that.
I'm hoping I can ask one more compelling question because we're up against time. You had mentioned work with payers. How may these devices impact the post-approval space? And will payers require use of the device in the access space if approval happens based on the data from the device?
Well, in general, the payers, at least in Europe, used to consider the same primary outcome than the trials. So they, when DHTs will be mostly used in the trials, it's very likely that they could become much more easily what we call a companion device, right? With the drug. This being said, I mean, if we want to be slightly provocative, when you look, for instance, in Duchenne, the number of drugs that are now either approved, either going to be approved, and if you consider to use all these drugs together, it becomes an incredibly expensive story, right? I mean, gene therapy is priced like 3 million. I mean, any exon skippers and our drug, the minimum is $200,000-$300,000 per year. So it means that from a payer perspective, if you're ready to invest so much money into a drug, you could invest 2% or 1% of this money into a good data collection and evaluation of being sure that what you do is the right thing to do. Also from a patient's perspective, I mean, I'm not aware of a single drug in the world that has 100% of responder. I mean, all drugs may have non-responders. And as a patient, as a potential patient, I would be happy to know if I'm a responder or not, because if I'm not a responder, please do not expose me to a drug that doesn't work on me. Maybe it works on a population, but maybe it doesn't work on me. So I think that when it comes to post-approval, it's the interest of everyone, the payers, the society, the patients themselves, and also the pharma company, because to deliver a drug on the long term, if it doesn't work, doesn't give a good image of the drug and a good understanding of the drug. So I think that we should be collectively ready to invest a little bit more in the understanding of the post-approval effect of the drug and to let it only to pharma company and telling the pharma company, you need to have registries and long-term studies is probably a little bit dangerous if we want to be entirely objective. So the short answer is today, not so much. Tomorrow, I think it will become more and more a possibility because again, many payers follow the primary endpoint of the trials. And as a vision, I think that having a complete independent way to assess the patients in the post-approval would make me comfortable as a doctor, because it's terrible to tell a patient, well, I think you are a non-responder. Maybe we should better stop this drug in you. This is not something a patient is happy to hear. So if we can, before starting a very expensive treatment, tell the patient, well, we have 10% non-responders. It's better if you're not exposed to a drug for which you are non-responder. So let's try to have the most objective and sensitive way to figure out if you have a benefit of this drug. And then if you do, so far so good. But if you don't, maybe it's your interest also to stop taking this drug. So I think again, it's more like a collective understanding of how to act the most efficiently for everyone.
Thank you, everyone. I appreciate all of your insights. This has been wonderful.
Yes, thank you. We want to give, again, another thank you to all of our speakers and our presenter. We do have another webinar coming up in June, Hidden in Plain Sight, Autosomal Dominant Tubulio. I'm not even going to try to pronounce it. Just take a look there. Please join us. We would love to have you. We will be sharing the video after the call, and there's a question about sharing slides, and we'll follow up with that and any other questions that have come through that we haven't had time today. So again, thank you very much for joining us, and have a great rest of your day.
Thank you. Bye-bye. Thank you.
Thank you.
Bye.
Yeah.